Dear students, today we will talk about histology of the organs of urinary system. So, urinary system includes the following organs. It includes 2 kidneys, it includes 2 ureters, urinary bladder and urethra. Kidneys, they produce urine, so they provide the main function of urinary system. They release waste products from the organism via production of urine. And ureters, urinary bladder and urethra, together they form urinary tract, which provides transport of urine from kidneys to external environment. So, main kidney functions are production of urine by filtration of blood plasma. Also, they regulate blood volume, blood pressure, because they release extra volume of fluid. Also, they regulate osmolarity because they release some electrolytes, and this way they regulate electrolyte balance, and also acid-base balance because they release some hydrogen ions and another ions, and according to this, they regulate acid-base balance of blood. And also they eliminate waste products, and in the main kidney function, they release waste products into external environment. So urine contains waste products, and also it contains extra water volume which is released from blood. It contains different electrolytes which should be released from the organism, and also acids or bases. And according to this, kidney regulate blood volume, pressure, osmolarity, electrolyte balance, and acid-base balance. And let's begin from the kidney anatomy. Kidneys, they include 2 main layers. Outer layer is cortex and inner one is medulla. Cortex is darker and medulla is pale. This is a result of differences in blood supply, and later we will talk about this. So here we can see tissue of kidney, and also there are hollow structures inside the kidneys. At the beginning of the urinary tract, there are calyces, minor and major, and renal pelvis, which is continued by the ureter. So it's urinary tract which begins inside the kidney. And tissue of kidney is divided into cortex, its outer layer, and medulla, its inner layer. Here we can see outer layer, it's cortex, it's darker than medulla. Medulla is pale and it's the inner layer. And medulla is formed by renal pyramids. Here we can see renal pyramids, and their number may be different from 5 till 12. It's individual anatomical variability number of the renal pyramids. And renal pyramids, they are main structures which form medulla of kidneys. And here we can see that renal cortex, it enters between the renal pyramids. It forms layers which separate neighboring renal pyramids, and those layers of cortex between the renal pyramids are called renal columns. So renal columns are inclusions of cortex inside medulla. And here we can see that medulla also enters in the cortex, and there are special extensions of medulla which enter in the cortex, and they are called medullary rays. They have shape alike the rays, and they enter in the cortex. So both cortex and medulla, they have structures which enter in each other. Cortex enters in the medulla as a renal columns, and medulla enters in the cortex as a medullary rays. And entire kidney is covered by the renal capsule. It covers kidney, it's dense fibrous connective tissue which covers renal tissue. So extensions of cortex, renal columns, they divide medulla into renal pyramids, or they separate neighboring pyramids. And here we can see that pyramid and region which surrounds this pyramid is called renal lobe. Here we can see region of the kidney. Its anatomical unit of the kidney, it's called lobe. It's renal pyramid and neighboring cortex. Lateral sides of the renal pyramid are covered with cortex, and basis of this pyramid also is covered by cortex. So here we can see renal lobe, its cortex, its renal pyramid which forms medulla of the renal lobe. And each renal lobe, each renal pyramid is opened into the minor calyx. Here we can see minor calyx which collects urine from the renal lobe. Here we can see point of pyramid. It's called papilla and it's nested in the cup in minor calyx. Here we can see scanning electron microphotograph and its apex of the renal pyramid, its appearance of it, and it's located in the minor calyx. And we have some minor calyxes, their number corresponds to the number of the renal lobes, and 2 or 3 minor calyxes, they form major calyx, bigger one which collects urine from these minor calyxes. Major calyxes, they form renal pelvis, common cavity of the kidney which collects all urine from all renal lobes, and it's continued by the ureter. It's extrarenal urinary tract, beginning of the urinary tract. So here we can see hollow structures inside the kidneys and also tissue of kidney, and kidney is considered as a parenchymal organs because it has parenchyma and stroma, and later we'll talk about this. And functional units of kidney, they are special tubules which are called nephrons. Here we can see nephrons. So nephron is the structural and functional unit of the kidney. And we have more than 1 million of nephrons inside each kidney. Here we can see nephrons. And also we have collecting ducts or collecting tubules and several nephrons. Some nephrons are opened in one collecting duct. So here we can see also another tube-like structures inside the kidneys which collect urine from some nephrons. And it's another system of the renal tubules. And here we can see collecting tubules, and they are opened into the minor calyxes. They have some orders of branching, and here we can see renal tubules, and regions where they enter in the cortex, they correspond to the medullary rays. So inside the medullary ray we have collecting tubule, and also it collects urine from some corresponding nephrons. And region of the kidney which corresponds to the one medullary ray to the region which corresponds to one collecting tubule is called renal lobule. This region is called renal lobule. So renal lobule, it includes nephrons, a region which contains nephrons which are opened in one collecting tubule. So some nephrons which are located here in this region, in the region of the renal lobule, they are opened in one collecting tubule or one collecting duct. And this collecting duct collects urine from all nephrons which are located in the renal lobule. And here we can see renal lobule. So renal lobe, it contains some renal lobules. Lobules are smaller than renal lobes. And lobules, they, in the center of the renal lobule, we have collecting duct, and also nephrons which are opened in this collecting duct. To summarize this, lobule consists of a collecting duct in the center and all the nephrons that open into this duct. And between the lobules we have layers of connective tissue with blood vessels which separate neighboring lobules, and about this we will talk a little bit later. So here we can see that collecting ducts, they are opened into minor calyxes at the top of the pyramid. And here we can see openings of the collecting ducts. It's point of the renal pyramid, and there are openings, and it's the region where urine is released into the minor calyx. So the main structures of the kidney are two systems of tubules. We have nephrons and collecting ducts. Here we can see some nephrons which are opened in one collecting duct, and they are main structures. So generally accepted opinion that the main structural and functional units of kidneys are nephrons, but also important structures of kidneys are collecting ducts which also participate in the formation of urine. But they aren't parts of nephron because some nephrons are opened into the same collecting duct. So here we can see distinct nephrons, and they are opened into this tubule. Here we can see nephron and its collecting tubule or collecting duct, and they are epithelial structures, and they form parenchyma of the kidneys. Kidneys are parenchymal organs. So parenchymal organs, they have two components, parenchyma and stroma. Parenchyma is specific part of the organ. For example, glands, they also have parenchyma and stroma, and parenchyma is different in every organ, and usually it's composed of epithelial tissue, but there are some exceptions. So specific part of kidneys, which provides main functions of kidneys, its system of two ducts, nephrons, and collecting ducts, two systems of ducts. They form parenchyma, it's epithelial part of the kidney. But also we have connective tissue which fills the spaces between the tubules, all those epithelial structures. It's loose fibrous connective tissue, and also there are blood vessels, and we call this stroma of kidney, or in the kidneys, stroma has special name which is called interstitium. So it's interstitium or stroma of the kidneys, which fills the space between the renal tubules. And let's begin from the structure of nephrons, which are structural and functional units of kidneys. So here we can see a diagram which shows the structure of nephron. And you should know that collecting duct isn't a part of the nephron. It's also important structure of kidney, and later we'll talk about its structure. So nephron. Nephron, it has two principal parts. It's renal corpuscle or renal body, and renal tubules. Here we can see renal tubules. So beginning of nephron, it's renal corpuscle, and it's continued by renal tubules. Corpuscle and renal tubules both are formed by epithelial tissue, and there is a same cavity, common cavity, which begins as a cavity of renal Bowman's capsule, and it's continued inside renal tubules. And this tubule is opened into the collecting duct. So renal corpuscle is capillary glomerulus, here we can see it, and Bowman's capsule, which surrounds this glomerulus. And renal tubules, they may be convoluted, they may be straight, they may be thick, and also they may be thin. And also they may be proximal, which are located near the renal body, near the renal corpuscle, and distal, which are located near the collecting duct. So here we can see kinds or regions of the renal tubules. Here we can see that nephron, it has one tubule, but it has some regions which have own distinct names. So here we can see proximal convoluted tubule. It's the first tubule which opens directly from the Bowman's capsule. It's proximal convoluted tubule. And it's continued into proximal straight tubule. Here we can see it. It's continued by thin limb or thin tubule, which has descending and ascending limbs. Then we have distal straight tubule and distal convoluted tubule. So here we can see beginning and the end of nephron, it's straight, it's convoluted proximal and convoluted distal tubule. Proximal convoluted tubule, it's also called PCT, and distal convoluted tubule also is called DCT. And deeper, they have proximal straight and distal straight tubule, and between them we have thin tubule. So proximal convoluted, distal convoluted, proximal straight, distal straight, and thin descending and thin ascending tubule. So there are main renal tubules, and in the correct order we have proximal convoluted tubule, proximal straight tubule, thin descending tubule, thin ascending tubule, distal straight tubule, and distal convoluted tubule. And here we can see main tubules of the nephron. And proximal straight, thin, and distal straight tubules, they form the loop of nephron or loop of Henle. It's a part of nephron which mainly enters in the medulla. Convoluted tubules and renal body, they mainly are located in the cortex, but this part, straight tubules as well as thin tubule, they enter in the medulla of kidney. They are located deeper. And here we can see loop of Henle. It's the part of nephron which enters in the medulla. And here we can see renal pyramid and cortex. So it's renal lobe. It's drawing of histological microphotograph. And here we can see kidney. Also it's drawing, but we have here bigger magnification. And we can see distribution of different parts of nephrons in different parts of kidney. So cortex, it contains renal corpuscles or renal bodies. And also it contains proximal and distal convoluted tubules. They form renal labyrinth in the cortex of kidney. Here we can see numerous sections of proximal and distal convoluted tubules. And deeper, we have in medulla, we have parts of loop of Henle. There are straight tubules, proximal straight tubules, and proximal and distal straight tubules, as well as thin tubules. So here we can see straight tubules mainly in the medulla of kidneys. And also medulla contains collecting ducts, but collecting ducts, they are going to the cortex, and here they form medullary rays with the neighboring parts of nephrons. So here we can see medullary rays, regions where medulla particularly enters in the cortex. So cortex contains the following structures: renal corpuscles or renal bodies, convoluted proximal and distal tubules, and sometimes it contains the initial portions of the collecting ducts and the upper portions of the straight tubules. And medulla, it contains straight proximal and distal tubules, thin descending and ascending tubules, and lower distal portions of the collecting ducts. So it's the distribution of the parts of nephrons and collecting ducts in the cortex and medulla of kidneys. And here we can see cortex of kidneys. Here we can see in the cortex renal bodies or renal corpuscles is glomerulus and Bowman's capsule. And also here we can see numerous sections of proximal and distal convoluted tubules. And here we can see round sections, its transverse section, and also elongated sections. They are oblique. And there are medullary rays, inclusions of medulla inside the cortex. And deeper, we have straight tubules, proximal and distal, collecting tubules or ducts, and also there are thin tubules in the medulla of kidneys. And it's the point of the pyramid, apex of pyramid, and it's openings of the collecting ducts on the apex of pyramid. And here we can see that it's covered with transitional epithelium, which is characteristic for the urinary tract, organs of the urinary tract, and later we will talk about them. So let's talk about structure of the parts of nephron and also about their functions. And we will begin from the beginning of the nephron. It's renal body or renal corpuscle, and it has two principal parts. It's glomerulus, it's group of the capillaries, which are formed by two arterioles. One carries blood to the glomerulus, another carries blood away from the glomerulus. And also there is a Bowman's capsule. And main function of the renal corpuscle is filtration of blood plasma and formation of primary urine. So renal corpuscle is tuft of the capillaries, it's glomerulus. Here we can see two arterioles. Efferent arteriole carries blood to this glomerulus, and efferent arteriole carries it away. So here we can see, and there are loops of the capillaries, and they are covered by the inner layer of Bowman's capsule. So here we can see Bowman's capsule, it has outer and inner layer, and inner layer it covers capillaries, and it's a place of blood plasma filtration. And fluid filters from the capillaries to the capsular space and then flows to the renal tubule. And here we can see very important feature of the blood supply. First of all, we have two arterioles. One is efferent arteriole, which carries blood to the capillaries. And also we have efferent arteriole, and all capillaries, they are located between two arterioles. So it's the exception from the generally accepted rule, which says that all capillaries are located between artery and vein, or between arteriole and venule. And its exception because capillaries are located between two arterioles, and this efferent arteriole later will be divided into secondary capillary network, which is located near the tubules, and it participates in the reabsorption. So two arterioles and capillaries between them, it's called rete mirabile arteriosum, or wonderful network of the capillaries between two arterioles. And another feature of this blood supply of this capillary bed is that arterioles, they have very different diameter. Here we can see efferent arteriole, which carries blood to the capillaries. It has bigger diameter, much bigger diameter than efferent arteriole, which collects blood from the capillaries and carries it away. So this difference of diameters, it results in the formation of hydrostatic pressure in the capillaries, because this arteriole can carry away less blood than this arteriole carries to the capillaries. That's why under this pressure, some amount of fluid should go somewhere, and it goes through the wall of the capillaries to the cavity of Bowman's capsule, and its formation of primary urine. So here we can see that hydrostatic pressure provides filtration in this region. So let's talk about structure of the Bowman's capsule. It has two layers, visceral or inner layer, and outer, its parietal layer. Both layers, they are formed by epithelium, and this epithelium is classified as simple squamous. But inner layer of this Bowman's capsule, it has specific cells, and they are called podocytes. Here we can see them. They also may be classified as a simple squamous epithelium, but they have some features of their structure because they provide specific functions. So here we can see podocytes. They have star-like shape. They are branched cells, and they have primary and secondary processes. Primary processes or feet processes, and secondary. Primary like feet and secondary like fingers. That's why podocytes were called podocytes. Podos, it's feet or leg. So here we can see podocytes which surround the capillary. Here we can see capillary of the glomerulus, and two podocytes with primary or big processes and secondary or small processes. They form neighboring podocytes. They form interdigitations. Here we can see interdigitations. What is interdigitation? Digit, it's finger. So interdigitations, it's pattern how processes of podocytes are arranged. And here we can see this pose when fingers are crossed in this way. And in the same way, secondary small processes of podocytes are arranged, and there are gaps between them, and those gaps between the secondary small processes of podocytes are called filtration slits. They are located here between the so-called fingers of podocytes. So here we can see pattern how they are arranged. And here we can see capillaries. It's outer surface of the capillaries, and they are completely covered by podocytes. Here we can see cell bodies of podocytes, and primary processes of podocytes, and secondary processes, thin processes, and here we can see interdigitations of them. And also here we can see scanning electron microphotograph. It's glomerulus, entire glomerulus, and there are numerous capillary loops here. Their relief is seen here, and there are cell bodies of podocytes, and their primary processes are well distinguished here, and also there are secondary processes which completely cover the surface of the capillary, so surface which is exposed into the Bowman's capsule, and they provide filtration, selective filtration of blood plasma. And on another side, in the capillaries, we have endothelial lining, special simple squamous epithelium. It's called endothelium, and cells are called endotheliocytes. And endothelium of the capillaries, it has pores, gaps or openings in the cytoplasm of the endothelial cells, and it increases permeability of the capillaries. Type of the capillaries, it's fenestrated type. On this picture, only one capillary loop is shown, but also there are numerous capillary loops in the glomerulus. And here we can see also mesangial cells which fill the space inside the capillary loops. It's mesangium. Mes, it's between angi, it's blood vessels, so there are special mesangial cells between the blood vessels and inside the capillary loops. So here we can see special structure which is called filtration barrier, and let's look what structures does it include. Here we can see capillary, and capillary is of fenestrated type, so there are openings, pores, special pores in the endothelium, but basement membrane is continuous. And it's covered by podocytes. Here we can see podocytes, and they completely cover the wall of the capillary. And here we can see inner layer of the Bowman's capsule. It's composed of podocytes, outer layer, and also it's beginning of the proximal convoluted tubule. And all these structures, they have basement membrane. Here we can see basement membrane. And all basement membranes, typical, they have two layers. Inner or upper, it's lamina lucida, which is located near the epithelium. It's lighter layer, light layer, and outer layer or deeper layer, it's lamina densa or dark layer. So light is located right under the epithelial cells, and lamina densa is located deeper. So here we can see Bowman's capsule, and podocytes, they also have own basement membrane. And here you can see capillary loop, and it has endothelium, simple squamous epithelium, and also it has own basement membrane with two layers, lamina lucida and lamina densa. And in many organs, we have a gap between two basement membranes, one of the capillary and another of another epithelial structures. And this space usually is filled with loose fibrous connective tissue, but not in the kidneys. And in the kidneys, two basement membranes are fused together, and it's needed to reduce this distance between blood and urinary space in the Bowman's capsule. So two basement membranes are fused, and as a result of this, they form common three-layered basement membrane, which has two lamina lucida, light layers, and two lamina densa. They are fused and they form common dark layer, which is located inside. So two light layers and one dark layer is inside. It's structure of the basement membrane of the renal corpuscle. So common basement membrane forms to reduce distance between blood and urine, and to provide permeability of the filtration barrier. And blood comes here from the efferent arteriole. It goes to the capillary loops, and from the capillary loops it's carried by efferent arteriole, and difference in their diameters results in the formation of hydrostatic pressure, and under this pressure some amount of blood plasma is filtrated by this barrier. So filtration barrier includes the following structures. It's capillary, wall of the capillary, which includes endothelium, simple squamous epithelium with fenestrations or with pores. Also basal lamina or common three-layered basement membrane, which is a result of fusion of two basement membranes, and also podocytes. So filtration barrier forms, it contains three layers. and it provides filtration of blood plasma, and about 100 to 180 liters of primary urine is produced per day. And here we can see filtration barrier, it's endothelium, common basement membrane, and podocytes, three main components of filtration barrier. And here we can see endothelium with spores, it's common basement membrane, three-layered, and small processes of podocytes, and between them we have special diaphragms which close these spaces, and they provide selective filtration of blood plasma and formation of primary urine. And here we can see capillary loop. It's urinary space of Bowman's capsule and its filtration barrier. And let's look at the selective permeability of this filtration barrier. So first of all, blood comes here. It's continuous circulation of blood in these capillary loops, and blood contains formed elements. They don't pass through the filtration barrier, so erythrocytes in normal condition are never present in blood, and in the if you see in your patient's urine, erythrocytes are present, which arise from the glomerulus, it says that it's increased permeability of this filtration barrier, that this filtration barrier was broken. But leukocytes, they have amoeboid movements, and they can pass through other barriers, and they can be present in urine, but only single leukocytes may be present. And if there are numerous leukocytes, it says that it may be inflammation in kidneys. So formed elements, they are too big to pass through the filtration barrier, and they never pass through the filtration barrier in the normal condition. And let's look at another structures and substances. Which substances can pass through the filtration barrier, and what is the difference between blood plasma and primary urine? So first of all, we have blood here. We have water, which is a main component of blood plasma. And here we can see that some amount of water comes here, and some amount goes to the efferent arteriole, but in other parts of water, they pass through the filtration barrier easily, and they go to the proximal convoluted tubule. And we have about 100 to 180 liters per day, and here we can see how huge is amount of water in the primary urine. Also, there are waste products in blood, and main function of kidneys is to release waste products in urine. So here we can see some waste product comes here, and it passes through the filtration barrier and goes to primary urine. Also salt, sodium chloride comes here, and its ions, usually they are separated from each other, and they also easily pass through the filtration barrier. And let's go to the organic substances. It's glucose, sugar. So glucose comes here, and it's a small molecule, it's monomer, and it passes through the filtration barrier. So primary urine may contain both salts and sugars. Next, it's polypeptide, and proteins may be of different size and big molecules, big protein molecules. They cannot pass through the filtration barrier. It isn't permeable for big molecules of proteins, and this molecule is carried by blood to the efferent arteriole. But small polypeptides, they can pass through the filtration barrier, and they enter in the primary urine. So it's selective permeability of the filtration barrier, and it depends on the size of molecule. And amino acids, small monomers, they can pass through the filtration barrier. So an amino acid comes here, and it easily passes through the filtration barrier. So what substances may be present in primary urine? It's water, it's waste products, different salts and ions, glucose and another monosaccharides, small protein molecules and amino acids, those components which also are present in blood plasma. What are the differences? In blood plasma, we have formed elements, they don't pass through the filtration barrier, and also big protein molecules, they also don't pass through the filtration barrier. All other substances are present in the primary urine, so primary urine is very similar to the blood plasma. And here we can see a histological drawing of histological slide, and here we can see Bowman's capsule, its outer and its inner layer, which covers capillary loops. And here we can see capillaries and its mesangium, its efferent and efferent arterioles, and also its beginning of the proximal convoluted tubule and neighboring distal convoluted tubules and proximal convoluted tubules. And here we can see diagram and drawing of histological slide. And it's microphotograph of the glomerulus and renal corpuscle. And here we can see glomerulus, and those numerous cells are cells of the capillaries, cells of the mesangium, which is located between the capillaries, and also there are numerous nuclei of podocytes. But we cannot distinguish between them, so common cell mass is seen here as a glomerulus. And its Bowman's capsule, here we can find simple squamous epithelium of the parietal layer of Bowman's capsule and its beginning of the proximal convoluted tubule. Here we can see it. And now we are going to the structure of the renal tubule, and you know that it contains proximal convoluted tubule, proximal straight tubule, thin tubule, thin descending and thin ascending limbs, distal straight tubule, and distal convoluted tubule. And collecting duct, it's not a part of the nephron, so it's distinct structure, but also it participates in urine formation. And main processes which take part in the renal tubules are reabsorption, uptake of some substances which are needed for one's organism needs back to the blood from primary urine, and also secretion of some substances, active secretion to the urine. And renal corpuscle, it forms primary urine by filtration, and renal tubules together with collecting duct, they form secondary urine by reabsorption and secretion. So here we can see proximal convoluted and proximal straight tubule, and on this picture we can see here that they are shown as a single tubule because they have the same epithelial lining and the same function. It's thin tubule, here we can see it, and distal straight and distal convoluted tubule, they also are shown as a single tubule to simplify this picture, and they have the same epithelial lining and the same function. And it's collecting tubule, so it's the main histological structures of nephron. And let's talk about them. So here we can see proximal tubule, convoluted and straight, thin tubule, distal tubule, also straight and convoluted, and collecting tubule. And in tubules of nephrons, we have only two kinds of epithelia. All epithelia are here simple, only one layer of cells is present. And in proximal and distal tubules, we have cuboid epithelium, so epithelium is simple cuboid. And in thin tubule, we have simple squamous epithelium. So simple cuboid, simple squamous, and again simple cuboid epithelium. And in the collecting tubule, in the upper parts we have simple cuboid epithelium, and in the lower parts we have simple columnar epithelium because diameter becomes to be bigger and cells are taller there. And what is the difference between proximal and distal tubules? Both tubules, they have simple cuboid epithelium, and they have very specific structures. There are basal foldings or basal striations, special foldings of the lower membrane of the cell, and between those foldings there are numerous mitochondria. Why do we have mitochondria there? Because in the proximal and distal convoluted tubules we have very active processes of reabsorption of ions, and there are pumps are working and they require energy. So energy is produced in mitochondria, and that's why there are numerous mitochondria near the basal pole. Because, for example, sodium-potassium pump is located near the basal pole of the cell and it's actively working here, and mitochondria, they provide energy for the sodium-potassium pump. That's why active cells, which provide active transport, they are cuboid, they have bigger volume and bigger size. And thin tubule, it provides passive transport of water, and it has a squamous shape because it doesn't provide active transport. And the difference between cells in the proximal and distal tubules is present on the apical surface of the cells. In proximal tubules, we have microvilli, which form the brush border here. And in distal tubules, we haven't brush border. It's because of functions of these tubules. In proximal tubules, we have active reabsorption of the organic substances. You know that there are proteins, there are amino acids, there are sugars, and they should be reabsorbed by the cells. And also about 85% of water is reabsorbed in the proximal tubules. So we need to increase surface area of these cells, and that's why there are microvilli which form brush border, which is alike brush border in the intestines. So the same processes which remind us of intestinal absorption of nutrients, they take place in the proximal tubules. And in distal tubules, we don't have organic substances, and that's why they have a smooth surface of the apical pole, but they provide active transport of ions. That's why they still have mitochondria and basal folds near the basal pole. So let's look at all those tubules. So proximal tubule, it has simple cuboidal epithelium with microvilli with brush border, which increases surface area of the cells, and it provides absorption of the nutrients into the bloodstream. And they form microvilli, brush border, and this way proximal tubule absorbs about 85% of the water. You remember that about 100 of liters per day of primary urine is produced, and about 85 liters of those water goes back to the blood vessels in the proximal tubules, and about 15 liters per day are going to the following tubules, where also reabsorption of water takes place. Also, sodium goes to the blood vessels and it's reabsorbed, and 100% of glucose and amino acids, they are reabsorbed in the proximal tubule. So all nutrients or important organic substances are going back to blood in proximal tubules, and it's called reabsorption. Here we can see what substances are filtrated and which substances are reabsorbed into the secondary capillary network. Here we can see blood circulation in the proximal, in the capillary loops, in the glomerulus, and also near the tubules we have secondary capillary network, and here we can see also blood circulation. And some substances are going to this blood, and let's look how reabsorption is provided. So first of all, water. Some amount of water is filtrated here, and usually 85% of water, they are going back to the bloodstream, and it takes place in the proximal tubules. So this drop, it shows us the water, and it goes to the blood. But some amount of water, 15% from the proximal tubules, goes to the following parts of nephron, and about 2 or 1.5 liters per day of secondary urine are released to the external environment. So some amount of water isn't reabsorbed, and it goes to the following renal tubules. Also, we have here waste products. They are filtrated and they enter into the blood plasma, they enter into the primary urine, and they pass through the proximal tubule without any absorption. sugar, glucose. Here we can see a molecule of glucose. It also is filtrated. It goes to the primary urine, and here it goes back to the blood because cells of the proximal convoluted tubule, they uptake this glucose and its reabsorption of glucose, and it goes to the blood. But in some conditions, for example in diabetes mellitus, or when we eat too many sweets, level of glucose in blood is increased. And let's imagine the case where when number of glucose molecules in blood is increased, and all those molecules may be filtrated into primary urine, and here we have 10 molecules or 10 millimole per liter, and cells of proximal tubules, they have some capacity of reabsorption. So 9 of those molecules are going to the blood. But when we have too high level of glucose in blood and too many molecules of glucose here, some molecules of glucose aren't absorbed, they aren't going to the blood, and they are going to the following tubules of nephron, and they enter in the secondary urine. And its mechanism of formation of glucosuria. Usually, its presence of glucose in urine. Usually, it occurs in diabetes mellitus, but also it may occur as a transitional sign. For example, when you eat too many sweets, and this glucose sometimes may be present in urine. So it goes to the following renal tubules. Polypeptides, small polypeptides, they may be filtrated via this filtration barrier, so they pass through the filtration barrier, and also they are reabsorbed in the proximal tubules. First of all, they are divided into amino acids. There are some enzymes here, and only after that they are absorbed by the cells of proximal tubules, and after that they are going to the capillaries. And here we can see this absorption. And salt, here we can see salt, and it comes here. Some amount of salt is reabsorbed in the proximal tubules and it goes to the blood. And here we can see that absorption of salt, of sodium and chlorine, they are absorbed separately from each other, and they are going to blood. But in other cases, some amount of salt goes to the following renal tubules, and especially distal convoluted tubules and distal straight tubules, they are involved in the reabsorption of salts. So here we can see main substances, and we can see that water, waste products, and some ions are going to the distal tubules. In thin tubules, it isn't involved in the active transport. It has simple squamous epithelium, and that's why it provides passive transport of water. Passive absorption of water takes place here. And we are going to the distal tubule. So epithelium is low cuboidal without brush border, but it has numerous mitochondria, and there are sodium-potassium pumps on the basal surface. This tubule isn't permeable for water, so solution comes here, and only salts are going to the interstitium. There is sodium-potassium pump, which is controlled by hormone aldosterone, which is produced in the adrenal glands. So aldosterone activates absorption of sodium, and it goes back to the blood vessels, and it's sodium-potassium pump, which is located on the basal surface of the cells in the distal tubules. And also they provide final absorption of the salt, and also they provide regulation of the water and acid balance because of release of hydrogen ions and ammonium ions into the lumen. So distal tubule participates in the secretion also not only in the reabsorption. And after absorption of salts, urine goes to the collecting tubules and ducts. They lie in the medullary race, and they are open to the minor calyx. And in the upper portions, they have simple cuboidal epithelium in the smaller ducts, and in the larger ducts, there is columnar epithelium. here we can see collecting duct in the smaller ducts in the upper parts of the kidneys. In the cortex, they have cuboid epithelium. In the lower parts, they have simple columnar epithelium. And they have two types of cells. There are light and dark cells. Light cells, they provide transport of water, and they are controlled by antidiuretic hormone or vasopressin, which increases permeability of the cells to water. And water absorption is activated in the light cells of the collecting ducts. And water diffuses from their lumens into the hypertonic medullary interstitium. And later we'll talk about this, what is hypertonic or hypotonic. And also dark cells, they are able for secretion, and they secrete hydrogen ions, and also they produce ammonium ions, so they participate in the secretion, and this way they regulate acid-base balance. Here we can see collecting tubule or collecting duct, and there are light cells and dark cells, and those cells, they produce ions of hydrogen, so they decrease pH of urine. And now let's summarize structure of nephron and also functions of nephron, and let's discuss the process of the urine formation. So the process of the urine formation, it includes three steps. It's filtration, reabsorption, and secretion, and they take place in different parts of nephron. Filtration takes place in the renal corpuscle, and reabsorption and secretion, they take place in the renal tubules and collecting ducts. And result of filtration, which takes place in the renal corpuscle, is formation of primary urine, it's blood plasma filtrate, and result of reabsorption and secretion is formation of secondary or final urine. So reabsorption and secretion, they form secondary urine from the primary urine. And let's look which parts of nephron do provide different functions. So here we can see blood plasma, and here we can see its drops. Blue drops are showing us water, and there are salt ions, salt. And for example, this ratio 1:1, it shows us isotonic solution, isotonic according to the blood plasma. So primary urine is isotonic. This ratio of water and salt is the same. And it goes to the proximal tubule, and here we have reabsorption of water and also salts. So isotonic solution is absorbed. And isotonic solution goes to the thin tubule. In thin tubule, we have mainly absorption of water. It's permeable for water, and a little bit hypertonic solution because of bigger amount of salts. It goes to the distal tubule. In distal tubule, we have hypertonic solution, and some molecules of salt, ions of salt, they are going to interstitium because distal tubule isn't permeable for water, it's only permeable for ions, electrolytes. And those electrolytes are going to the interstitium, and they attract water from the proximal tubules and also from the thin tubules. They attract water to the hypertonic interstitium. Salty interstitium attracts water to there. And also here we can see salty interstitium because of reabsorption of salt, hypertonic interstitium in literal terms. And also it helps in the reabsorption of water in the collecting tubules, and water is reabsorbed in the collecting tubules as well. So here we can see result of this, it's hypertonic concentrated urine. It's secondary final urine. So primary urine is isotonic, it's similar to blood plasma, and aldosterone activates a sodium-potassium pump, which provides reabsorption of sodium in the distal tubules. So it's main place of reabsorption of sodium. And here we have reabsorption of sodium in distal tubules, and it helps in the reabsorption of water in another tubules in proximal, thin, and collecting tubules. So a hypertonic interstitium, it attracts water, and more active transport of water is present in proximal tubules, in thin and in collecting tubules. So reabsorption of water, it has three places. It's proximal tubule, and here we can see reabsorption of water in this case. Also thin tubule, because salt, it attracts water to the interstitium, and also it's collecting tubule or duct, and here we also have reabsorption of water, and three places of reabsorption of water, and one is the place of reabsorption of sodium. But also proximal tubule is also able for the sodium reabsorption, but main place of sodium reabsorption, it's distal tubule. So primary urine is isotonic because it has the same concentration of ions as in the blood plasma. This fluid, which is present in proximal tubules, also is isotonic because isotonic fluid is reabsorbed. In the thin tubules, mainly reabsorption of water takes place under influence of salty interstitium. So this urine is hypertonic after reabsorption of water. Because it's more concentrated than in the proximal tubule. In distal tubule, the absorption of salts takes place, it's active transport, and interstitium is hypertonic because of big amount of salts, and interstitium is hypertonic, but this content of the distal tubules after reabsorption of sodium is iso or even hypotonic, less concentrated urine, because huge amount of salts is reabsorbed. And after that, isotonic or hypotonic urine goes to the distal tubule, to the collecting tubule, and here we have again the absorption of water, and it goes to the interstitium, to hypertonic interstitium, which attacks water, and this urine after reabsorption of water is again hypertonic. So final urine is hypertonic, it's highly concentrated, and we have about 1.5 or 2 liters of urine per day of secondary urine, and it's 100 times less than amount of primary urine. So 100 times urine is concentrated with huge concentration of waste products, and all these processes, they take place in the tubules of nephron. It's called reabsorption. So here we can see the process of urine formation. And now let's go to another function of kidney. It's endocrine function of kidney, and it's provided mainly by juxtaglomerular apparatus. Here we can see glomerulus, which we already know, and some cells of it, and cells which are located near the glomerulus, they form juxtaglomerular apparatus. Juxta, it means near something. So juxtaglomerular apparatus, it's apparatus which is located near the glomerulus. And it's located between efferent and efferent arterioles, and it includes juxtaglomerular cells, also macula densa, and extraglomerular mesangial cells. So let's talk about them step by step. First of all, macula densa, it's a disc of tightly packed columnar cells in the distal convoluted tubule at the point near the vascular pole of renal corpuscle. So one of the loops of the distal convoluted tubule goes back to the renal corpuscle and is located between two arterioles. Here we can see this distal tubule, and sometimes it goes between the arterioles. And some cells of macula densa are modified. They are dark and they are columnar, and it monitors osmolarity of the fluid in the tubule lumen. So those cells, they are sensory cells, and they react to the concentration of ions in the secondary urine. And some of these cells are sensory cells when urine goes here, and those cells are sensory. And also we have juxtaglomerular cells. In the adventitia of the arterioles, we have smooth muscular cells which provide contractions, they provide regulation of blood flow in these capillaries, but some of them are modified and they contain granules because they are secretory cells. and they produce hormone renin. And here you can see hormone producing cells. So they receive information from macula densa, which reacts to the osmolarity of blood, and it sends information to the juxtaglomerular cells, and they produce hormone renin. And also third component of juxtaglomerular apparatus, it's extraglomerular mesangial cells. They are located, here we can see usual mesangium, which is located inside the capillary loops. But this triangle between two arterioles and macula densa is filled also by special cells. They are called extraglomerular mesangial cells, and they may produce renin when juxtaglomerular cells, main cells, are exhausted. So when these cells are exhausted, their function is replaced by these cells which are located near them. So there are third cells. They are extraglomerular mesangial cells, or sometimes they are called juxtavascular cells. And here we can see juxtaglomerular cells. They are located in the adventitia of arterioles, its macula densa, and extraglomerular mesangial cells. So what is the mechanism of the hormones of juxtaglomerular apparatus? Juxtaglomerular cells, they produce renin. And renin, it activates the reaction of transformation of angiotensinogen to angiotensin 1. It catalyzes this reaction. And after that, angiotensin 1 is converted to the angiotensin 2 by angiotensin converting enzyme. And it always is present in blood plasma, so only renin is needed to activate this chain of the reactions. And angiotensin 2 is active, and it causes vasoconstriction, constriction of the blood vessels, and it results in the increase in blood pressure. And also angiotensin 2 activates production of aldosterone by adrenal glands, and it activates sodium reabsorption. Sodium reabsorption results in the activation of water reabsorption, and it increases blood volume by absorption of water. Main results of the release of renin is vasoconstriction, which results in increase of blood pressure, and also aldosterone also increases blood pressure. And here we can see mechanism how kidneys regulate blood pressure. And also this mechanism has very big clinical significance, because if kidneys suffer from hypoxia, they, in simple words, they think that entire organism has low arterial pressure because small amount of oxygen comes to kidneys, and they produce renin in big amounts, and renin increases arterial pressure, and it results in the renal hypertonic disease caused by the renal diseases. It's endocrine function of kidneys. And now let's go to the blood supply and circulation of kidneys. Each kidney, here we can see it, it receives a renal artery, its branch of the abdominal aorta. Here we can see renal artery, and anterior and posterior branches arise from the renal artery before it reaches the renal hilum. Here we have renal hilum, and later it is divided into smaller branches. They are called interlobar arteries. We remember that pyramid and neighboring cortex, it's renal lobe, and between lobes we have interlobar arteries. And they achieve the margin between medulla and cortex, and here they form arcuate arteries. They surround renal pyramids, and two interlobar arteries from two sides together they form single arcuate artery. So it's anastomosis between two arteries, and it helps in the continuous blood circulation in the kidneys. So here we have renal artery, also anterior and posterior branches of the renal artery, and they form segmental arteries. Interlobar arteries here are present, arcuate arteries, and also here we can see arcuate arteries, interlobar arcuate arteries, and here we can see small ray-like arteries which are called interlobular. We remember that there are medullary rays they are in the center of each lobule, and between the lobules we have interlobular arteries which have radial direction. So we have renal artery, segmental arteries, interlobar, arcuate, and interlobular arteries. Interlobular arteries, they arise from the arcuate arteries, and they lie at the borders between neighboring renal lobules. Here we can see a medullary race inside each renal lobule, because in the center of the renal lobule we have collecting duct, and all region which corresponds to this collecting duct is renal lobule, and between the lobules we have interlobular arteries. And here we can see its interlobar artery, its arcuate artery, and interlobular arteries which have radial direction, and their branches are efferent arterioles which are going to the nephrons. Here we can see, and now let's go to the classification of nephrons, and later we'll back to the blood supply. So there are three general classes of nephrons. There are cortical subcapillary nephrons, intermediate cortical nephrons or typical nephrons, and also there are juxtamedullary nephrons which are located near the medulla. So typical nephrons, their straight tubules and thin tubule are going to the medulla. It's typical nephron. Also we have subcapillary cortical nephrons and juxtamedullary nephrons. It's border between cortex and medulla, and here we can see that they have different situation, different localization. So subcapillary cortical nephrons, intermediate, and juxtamedullary nephrons. If the renal corpuscle lies superficially under the capsule, and those nephrons are called subcapillary cortical nephrons, and their loop of Henle almost doesn't reach the border between cortex and medulla. So entire nephron lies in the cortex. In most nephrons, about 80%, the loops descend to different depths in the outer zone of the medulla, and they are called intermediate nephrons or typical cortical nephrons. And in other nephrons, which form about 20%, they are called juxtamedullary nephrons, and they are located so their renal corpuscle is located near the border with medulla and also convoluted tubules, and loop of Henle is very long and it achieves the deepest parts of the medulla of the renal pyramids. And here we can see that the renal corpuscles of the cortical nephrons are located in the cortex. Here we can see bodies of the subcapillary nephrons, and deeper near the medulla we have renal corpuscles of juxtamedullary nephrons. They are located near the medulla but still in the cortex. And about blood supply of the nephron. So we have efferent and efferent arterioles. Here we can see efferent arteriole which has bigger diameter, and efferent arteriole, and it's divided into secondary capillary network which surrounds tubules and also collecting duct. And after that we have small venules which collect blood from the secondary capillary network. So we have two capillary networks. One is between two arterioles, it's glomerulus, and secondary it's peritubular capillary network. Here we can see arcuate artery, it's interlobar arcuate artery, branch of arcuate artery, it's interlobular artery, it has radial direction, and branch of the interlobular artery, it's efferent arteriole, which goes to the glomerulus, forms glomerulus, and efferent arteriole carries blood away from glomerulus, and it forms peritubular capillary network which surrounds tubules, which surrounds collecting duct, and after that venous blood from the secondary capillary network, peritubular capillary network, is collected by the venules, and they carry this blood to the interlobular vein. And here we can see venous circulation which repeats arterial circulation. So interlobular vein which enters into the arcuate vein, and arcuate vein is continued by interlobar vein. And here we can see removal of venous blood from the kidneys. It's typical cortical intermediate nephron and its blood supply. And subcapillary nephrons, they also have this pattern of blood supply. But juxtamedullary nephrons, they have some features of blood supply. And here we can see juxtamedullary nephron. Two arterioles. First of all, let's look here. Efferent and efferent arterioles, they have the same diameter, and it results in the absence of hydrostatic filtrative pressure. So these arterioles and this glomerulus doesn't provide filtration. Filtration is provided by the subcapillary and typical cortical nephrons. But juxtamedullary nephrons, they don't provide filtration. But what is their function? They are special extra way of blood. Here we can see that efferent arteriole, it forms a secondary capillary network, but also it forms vasa recta or straight blood vessels. And here we can see that arteriole directly transits into the venule. Here we can see straight arteriole or straight artery or vasa recta, and it transits directly into straight vein or straight venule, and it enters into the arcuate vein. So it's special extra way for blood, and sometimes in some cases we should decrease intensivity of blood filtration to decrease volume of urine which is released, and in those cases blood goes through the juxtamedullary nephrons. It's extra way for the blood, and more volume of blood is saved in the organism. For example, after hemorrhages or when we suffer from dehydration, kidneys, they save water in the organism and they distribute blood, and blood goes through the system of circulation of juxtamedullary nephrons, and it's easy way for blood to pass directly from the arterial part of the circulation to the venous without any filtration. So it's the function of juxtamedullary nephrons. It's extra way of blood when we need to decrease intensivity of blood filtration. Efferent arterioles of juxtamedullary nephrons, they give rise to the numerous straight capillary loops. They are called vasa recta, which descend into medulla, and they enter into the arcuate veins. So here we can see blood supply of typical cortical nephrons, which include intermediate and subcapillary nephrons, and its blood supply of juxtamedullary nephrons. And now let's talk about histology of the organs of urinary tract. It includes renal calyces, minor and major, renal pelvis, ureters, two ureters, urinary bladder, and urethra. And they are hollow organs, they have cavity, and they have a wall which has layered structure. Those organs, they have typical structure and typical layers, which are a little bit similar to the organs of digestive system. So first of all, we have mucosa, which is lined with special transitional epithelium, or also it may be called urothelium. Under this epithelium, we have lamina propria, it's loose fibrous connective tissue. Deeper, we have submucosa, but we don't have muscularis mucosa, so border between them isn't clear. Deeper, we have muscularis, but muscularis, look here, the outer layer is circular and inner is longitudinal. It's different from the digestive system, where inner layer is circular and outer is longitudinal. And the outermost layer is usually adventitia, but in some places it's serosa. For example, near the bottom of the urinary bladder, it's covered with serosa, with peritoneum, so it may be particularly adventitia or serosa. And special feature of the urinary tract is the epithelium, and epithelium is transitional, so it's called urothelium, and it's stratified, it has more layers than one, and transitional epithelium transits from squamous to cuboid or even columnar according to the filling of the organs of the urinary tract. For example, urinary bladder, when it's filled with urine, wall of urinary bladder is stretched and epithelium also is stretched. Here we can see transitional epithelium. Superficial cells of transitional epithelium are called umbrellocytes because they are maybe opened like umbrellas. So let's look. When organ is stretched and wall of organ also is stretched as well as epithelium, and cells become to be squamous. And here we can see umbrellocytes, they are opened like umbrella. And in the resting condition, wall is relaxed and cells again become to be cuboid or even columnar. So here we can see how transitional epithelium works. And here we can see two conditions of transitional epithelium in relaxed and stretched condition. Here we can see transitional epithelium and cells in relaxed and stretched condition. And here we can see special umbrellocytes or dome cells which cover the surface of the transitional epithelium. In ureters, here we can see epithelium of ureter. It's transitional epithelium, it's lamina propria and submucosa. In the longitudinal layer of smooth muscles, and outer is circular, and the outermost layer is adventitia, so it's structure of ureter. Urinary bladder is similar to ureter, but there are some differences. Here we can see epithelium of mucosa, lamina propria of mucosa, submucosa. Deeper we have muscles, but there are 3 layers of smooth muscles, and layers are oblique in the urinary bladder because they form musculus detrusor vesicae, and it's needed to provide urination. And the outer layer usually it's adventitia, but bottom of the urinary bladder is covered with peritoneum, and there we have serosa instead of adventitia. And feature of the urinary bladder, it's triangle of urinary bladder where we don't have folds of mucosa. It's because of absence of submucosa. In those places, mucosa is attached to the muscularis, and there are no folds in the urinary bladder. Here we can see structure of urinary bladder. It's transitional epithelium, it's lamina propria, submucosa, it's a detrusor muscle or muscularis with 3 layers. They cannot be distinguished clearly, but they are present there. And its outer layer, it's in this case, in this region, it's visceral peritoneum, so it's serosa, but in another places there is adventitia. And another organ of the urinary system, the last part of the urinary tract, it's urethra, and it has also the same structure, which is similar to the urinary bladder and ureters. It's fibromuscular tube which connects bladder to external urethral orifice, but it's sexually dimorphic. In males, it's also organ of the male reproductive system, and its way of spermatozoa to the outer environment. In female, its organism, it's shorter tube, it's wider, and it's organ of urinary system only. In both in females and males, it's lined by a transitional epithelium, and the same layers of wall are present there, but transitional epithelium transits into stratified squamous non-keratinized epithelium, which transits into the skin. Here we can see female urethra. It's transitional epithelium in this region, but it transits into the epithelium of skin. And layers which are similar to the ureter are present under the mucosa. In male urethra, another layers are thinner. Main layer is epithelium with lamina propria, so male urethra contains mainly mucosa with epithelium and lamina propria, and it's surrounded by cavernous tissue which is rich with venous blood vessels which are filled with blood. So students, that's all, and thank you for your attention.